Hello, everybody. My name is Bouabid Badaoui. I'm professor of bioinformatics and genomics at Mohammed V University in Rabat, and I'm the coordinator of North Africa for Africa BP project. Today we'll speak about landscape genomics in Africa, and especially I will tackle the case of goat and sheep breeds in Morocco. So when some environmental changes happen, organisms respond via local adaptation, which consists of permanent changes to an external pressure, or via climatization, that consists of temporary changes. Organisms could respond by migration too, or they can eventually go extinct. Local adaptation, which interests us in this case, describes the adjustment or change in behavior, physiology, and morpho-anatomy of an organism to better fit its environment. And evidently, locally adapted populations are expected to exhibit higher fitness in their native habitats than population formals were. Identifying genomic changes underlying local adaptation is critical in addressing fundamental questions in evolutionary biology, and this is the scope of the studies I'm presenting here. The goat and sheep species in Morocco constitute an important case to study, as those species are well adapted to contrasted environment, mountains, plains, Sahara, plateau, etc. And sheep and goats are present in big number in Morocco, and they are living in different area of the country in terms of environmental factors. So here we will use landscape genomics to first figure out if there are selection signature linked to environmental variation in sheep and goats, knowing that most adaptive traits are affected by many segregating loci, and thus quantitative approach is needed, as we will see later. Second, we will try to answer the question, is there a congruence of selection signature in both species, goats and sheep, knowing that those two species are closely related and undertook similar demographic and domestication processes. This put on the table the old debate among scientists concerning the relative impact of stochasticity versus natural selection in speciation and diversification. And lastly, we will ask the question, or we will try to answer the question, are there any geographical barriers that contribute to the differentiation among each species population, knowing that Morocco has a long chain of mountains that cross the major part of the country. So, as an approach for landscape genomics, 160 sheep and 161 goats were selected to cover different environmental regions with different environmental variables in a surface of 450,000 kilometers squares in Morocco. The whole genome sequencing and variant calling of those animals' DNA showed the big genetic diversity for both species. 38.6 million variant for sheep and 31.7 million variant for goats. This high degree of genetic diversity suggests that there were no strong bottlenecks that occurred during those pre-devolution, and that the gene flow has been maintained. And of course, it may mean too that there will no a lot of selection by the human of those species. This weak population structure has clever advantage when dealing with landscape genomics, as the confounding demographic factors are less present in this case. So we can study landscape genomics with a lot of ease in this case. The whole genome sequences of 161 sheep and 160 goats were correlated to 10 environmental variables, including temperature, rainfall, solar radiation, etc. And this combination was done to detect selection signature and biological processes involved in local adaptation of those breeds. So if you ask a question how these 10 environmental variables were chosen at the beginning, 67 climatic variables were considered, and after that, a pairwise correlation analysis between all 67 variables were performed to detect highly correlated ones, and the 10 representative variables were kept to investigate signature of selection. As a result, here in an example, we have here in figure A, a Manhattan plot of XP cellular scores across autosomes in relation to the mean temperature of July. This peak here in chromosome 23 comprise missense SNP, which is very interesting for landscape genomics. So figure B, it represents geographical variation of this SNP. And as you may see here, the CC genotype is very present in the west part of Morocco, and the TT and TC genotypes are present mainly in the center and in the south of Morocco. Figures C shows the frequency of the SNP for a group of 20 sheep along the temperature gradient. We can see that the frequency of the C allele decreases as the mean temperature increase. And of course, this is what concerns one SNP was done for all the variants that were found in this study. In another study, we were able to combine genomics and spatial data to highlight geographical barriers that contributed to the differentiation between the breeds in Morocco. And as you may see here, there is a mountain chain here, it's Middle Atlas, that separates population of goats and sheep in two parts of the country. Generally speaking, more than 12,000 and 8,000 variants linked to local adaptation have been highlighted in sheep and goat respectively. This corresponds to 276 and 294 genes in sheep and goats respectively. Most of the variants detected were enterogenic, and those that were exonic, I mean, were rare. We got 68 in sheep and only 1 in goats. So, and most associations, associations between those variants and the environmental variable, most of these associations were with the variant and the annual temperature range, the rainfall seasonality, and the altitude. So, however, if we look to the overlapping between the variants and the genes between the two species, we can find that there is no overlapping of variants, 0, and 1 overlapping genes between the two species, goats and sheep. This demonstrates that there is distinguished adaptive mechanism to local environments between sheep and goats. Such diversity of adaptive pathways might result from a high number of biological functions involved in adaptation to multiple eco-climatic gradients. And this provides more argument for the role of contingency and stochasticity in adaptation rather than repeatability of the evolution processes. The variety of genetic conversions between goat and sheep could be due to many reasons. We can cite at least two. First, the diversity of gene networks related to biological functions required for adaptation. I mean that one trait could be attained by different network of genes because there is some redundancy of biological function within the gene networks. And second reason could be the level of genetic variation that could be responsible for the variety of genetic conversion between the two species. But anyway, adaptive convergence between the two species could happen via the three scenarios presented here. Let's start with the first scenario. Goats and sheep use different genes to produce different adaptive traits which serve the local adaptation, as there is a redundancy of biological functions required for adaptation, as mentioned before. Second, goat and sheep use different genes to produce the same adaptive traits. This is because there is different genes could produce the same adaptive traits as there is some redundancy for the function of genes. And the third scenario is that goat and sheep use the same gene to produce the same adaptive traits, as it is more likely the case of the common gene CA2 between the two populations, because in a precedent slide, it was highlighted that the common gene, the only common gene between common gene related to environmental factors between goats and sheep was CA2 gene. So to conclude, landscape genomics applied to whole genome sequencing data identified several sets of variants, genes, and biological processes involved in local adaptation to various eco-climatic conditions. Through a whole landscape genomics, we can highlight the genetic variation over environmental gradients. As we mentioned before, the most important environmental factors that were correlated with genomic variant were rainfall, seasonality, and temperature, and altitude too. There are different adaptive mechanisms to similar environments in sheep and goats, as we have seen before. The overlapping between the two species is very scarce, with no variant overlapping and one gene overlapped between the two species. This provides more argument for the role of contingency and stochasticity in adaptation rather than repeatability. And this is an old debate between scientists stating whether evolution is mainly based on contingency and stochasticity and or in repeatability. Thank you very much for listening.